Hi friends, so now I'm back with my next video, and in this video I'm going to talk about something very special, which is the Nobel Prize in Physiology and Medicine 2023. So as you know, and this is a very proud moment, that this prize is awarded to Katalin Kariko and Weissman for their development of vaccines against COVID-19. So, as you know, they are well deserved for this award because the pandemic and the vaccine was very important to save the lives of people. So, this is a very nice achievement for both of them. And in this video, I'm going to talk about the concept that they use to develop these vaccines. So, as the name suggests, they use the nucleosides based modification to develop this vaccine. What does that mean is, as you know, that in the human system and in the cell, there is this process of the genes in which the DNA get transcribed to mRNA and then which is transcribed to proteins. And what they used for their vaccines is mRNA. And this process is not something very new that they used. They were working on them from a long ago. So the process of vaccine development from mRNA started in 2005 and then 2008, and then the same process was used in 2020 to develop vaccine. Okay, and how did they use and what did they use? So as you know, mRNA is, so this process to transcribe is called transcription, and then this process is called translation. And if you know, the proteins are the major unit that gives a particular phenotype. So what they use is the mRNA vaccine because the DNA vaccine, this process of transcriptions occur inside the nucleus, and then this process occurs in the cytoplasm. So if we use DNA vaccine, then the vaccine has to go inside the cell and then in the nucleus, and then from here to cytoplasm and then in the nucleus. And this process require huge amount of pulse. Okay, but for the mRNA, since it doesn't have to go to the nucleus because the process occurs only the cytoplasm, so it's easy. But with everything there are pros and cons. So what happens is DNA is stable, pretty stable, but mRNA is very unstable. Okay, so what they used, like in 2005 when they started working with this mRNA vaccines, the problem is because it's very unstable, and the technique that they used is they took mRNA. So as you know, mRNA is a single stranded with different bases, which are mainly A, U, G, C in case of mRNA, and if it's DNA, it's A, T, G, C. Because in RNA, the T, which is thymine, is replaced by uracil. Okay, so this is the base, and what they did, because it's very unstable and it can be affected by different chemical reactions which occurs in the human or host body. So what they did is they used some lipids which we called as liposomes around the DNA, and then this, because this will prevent the degradation of RNA. So this prevents the degradation of mRNA, and then they transmit these into the host, which can be mice and human. But when they did this, like in 2005, when the Moderna company they started this technique, what they found is that they never initiated they were never able to build these vaccines properly. There were multiple reasons. First was because when they injected this mRNA vaccine, it generated the immune response, particularly the interleukins, and then so this is basically a synthetic DNA, and this gets degraded, and then the mice was never able to build the vaccination response. So they were very confused, like why it is happening. So they were never able, because these interleukins are very damaging, and so they are like pro-inflammatory cytokines. So they initiated the cytokine response, and which can cause tissue damage in the host. Okay, so what then they were confused what else they used, and what they found is the presence of tRNA in the human body. So if you know, in the host, in the human, there are basically three type of RNAs which are found: mRNA, tRNA, and rRNA. So if you know, mostly we talk about mRNA because this gives protein, and this tRNA and ribosomal RNA are playing some role in the translation. And what they found is that tRNA, which is present in the human body, sometimes it's modified. Mostly it's modified. Because if you know, this tRNA contain different secondary structures, right, and loops, and this is mostly modified, in which, what does that mean is, the uracil which is present is mostly present as pseudouracil, okay, which is, if it's U, then it's present in the modification form. And so what did they do? They thought, because tRNA is with this pseudouracil, it's much more stable and it is prevented from degradation. So they used the same technique, in which the mRNA that they used, instead of having AUGC, they modified this U to uracil, pseudouracil. Now it is a pseudouracil GC. So and then the liposome around it. So when they injected the host with this vaccine, with the instead of uracil, they have the modified base. They found that when they did this, the RNA that they get from this modified is much more stable than the normal RNA. So as I told you about that, the previous mRNA, it's very unstable, so it degrades very fast. But when they modified it to pseudouracil, the mRNA was pretty much stable, and then the host does not recognize this as foreign. So it did not build much pro-inflammatory cytokines. Okay. So this modification solved two problems. First, it made the RNA much more stable, and then it did not initiate the pro-inflammatory cytokines and prevented from the host damage. Okay. So this technique was something which was used in the development of the COVID-19 vaccine, and because the scientists were working on this technique, like the Moderna and BioNTech, they were working on this technique from long ago. That's why they were able to build the vaccine so fast, pretty fast. Like, we got the pandemic and in 1 year we had the vaccine. And that's why this discovery was very important for the Nobel Prize for the Katalin Kariko to appreciate them, and to for this big achievement in the science field. Thank you, and don't forget to like, share and subscribe my channel. Thank you.